Здравствуйте.
Hello. Thank you indeed for the opportunity to talk to you on this conference. I would like to talk to you about what we do in our lab, but I would like to start with the technical director for Google, Ray Kurzweil, who is very well known for the accuracy of his future predictions, and because of his high accuracy, he has also published a book with his predictions up to 2,999, as someone would potentially, hopefully be lucky to see. But let's look at the coming future. We would like to just understand what's going on, what will happen in the nearest 10 years, and looking at those coming 10 years, they have nothing to do with genomics. Forecasts the interpretation to get digital, the car driving to become autonomous, Earth to be covered by the internet cloud, and transformation of all the human knowledge into digital form, but there's no genomics. But if we look at 2029 to in 10 to 20 years. But according to his predictions, we see first steps in understanding how the brain works and how the body works to be programmed or forecasted by roughly 2036, 2037, when by using the biology approach, CRISPR or biology programming approach, to program the cells and thus understand how the brain works. In terms of science advancements that we work on in our lab, of course, I'm monitoring the Nobel Prizes, and I've joined Kurt in our forecast. I think that genome algorithms and cell programming will be the nominations for Nobel Prize in 2036. Why do we talk about this? The whole bioinformatics which is an applied method for analyzing what's going on inside the cell and molecular biology, is the technology progress, genome sequencing progress. This progress has happened in the beginning of 2021, and genome sequencing has triggered a number of experiments, and this progress, as you know, computer science obeys Moore's law, with doubling something together with a price reduction, or the memory size doubles. So the split here, the error here from the Moore's law, the departure from the Moore's law, happened 13 years ago. So currently, to sequence your genome can cost $200 or $300. So the decrease of cost is like 10 to the power of 5 or 6. And because of this technology, we've gathered a huge amount of data in the molecular biology. This big data is represented here as multidimensional arrays, and besides just DNA sequences, just the markers that of functional elements of the genome that talk to us about how the cells should work in this or that situation. We have a lot of different types of cells and tissues, and we have the same genome, the same DNA sequence or letter sequence. So this meta coding or other layers are coded, and we get those that information because thanks to experiments, and because there's a lot of information the human brain cannot analyze and understand all those all those connections and relationships, and of course we wanted to use AI and especially deep learning to analyze them. So bioinformatics is using deep learning with a delay, with a lag to all the other areas, because in the most available bandwidth goes to machine translation or image recognition. So in bioinformatics, we see the already working implementations. We take them and try to apply them to the genome. So convolutional neural nets have shown significant progress, significant promise in picture recognition, image recognition, RNN for language interpretation, language translation. And we use them to predict where this or that functional element would be found in the genome. We can regulate them, and they are the targets for the treatments, for the medical work. So we take those CNN blocks, we digitize DNA as a binary vector, we add a binary matrix, we add omics data, which is the data that are generated by the line of those experiments for genome sequencing, next-gen genome sequencing that I have just shown you. And by accumulating this all, we push them through CNN, RNN, to forecast, to predict where those functional elements are. This is like decrypting of the genome code. So one of the unknowns in the coding is the so-called secondary structures of the DNA. And also we predict the cancer genomes. Cancer is a genome sickness, as you know, and here's an example from the site of the cancer genome site, accumulating terabytes of data to show what works where. And to the right you have a picture showing you that during cancer you have breaks, duplications, translocations. And using the machine learning, we do our best to find the determining factors that will help us to see the most important impact of this or that factor to affect those experiments, and those are gathered from those omics data. And the next line of business for our lab is genetical analysis, and together with the most prominent Russian centers, we work as a consortium to analyze the genetics of heart diseases. This is a less known area because there's not enough money invested there, and what we see here is that we can create predictive systems based on ML that, as part of the personalized medicine, those predictions are done not only based on the clinical research and analysis, but also based on the person's genome, whether he is potentially susceptible to this or that illness or this or that scenario. So this science is being developed very fast. It's, I don't know if it's linear or to the power of 2, but with the COVID, we got a huge boost to this science, and because we understand that even with COVID, there are different levels of susceptibility, and we don't know why one patient would have a hard scenario, a tough scenario with COVID, and one light scenario. And those steps are done together with Russian Medical Academy for constant professional development. We are basing this on their data to forecast for anti-aggregation or anti-psychosis medications. And another line of research we have, together with the Central Medical Academy, with Dmitry Zateshikov, we work to launch a pilot project to research the lung hypertension based on the machine learning methods that will be able to base that on the omics experiments data to reduce significantly the cost of analysis and get that omics data for each and every patient that we can analyze and then create a predictive system to help the patient. Thank you for your attention, and I would be happy to answer the questions. I have a question to ask. Given the development in genomics, what is the chance to get the new types of medications that will be able to fight onco-illnesses or heart diseases? Well, the progress will start not from the medication. It will be about screening. Huge progress in oncology currently has to do with screening, because you can find cancer in the early stages, and thanks to a consortium that was accumulating those mutations for dozens of years. We see now that despite all the heterogeneity of cancer, we have common drivers for those mutations that we were able to find. And this panel for 10 to 13 genes, you can have a blood test, and with the first changes, with the first presence of some proteins in your blood, you can predict the oncogenesis with very high accuracy. And of course, the progress will be amplified by the personalized medicine, with the research companies accumulating huge amounts of data. And when we see one patient not being affected by this medication, but the other having very good results, so this compliance with the medical treatment is imprinted into the genome. And if the protein is mutated, then there's no connection. So this is the future. Not Ray Kurzweil, so I will not tell you when this will happen, but given the fact this is a lot of focus for 20 years in terms of cancer, I think it's in the coming future. In the West, heart diseases are less prominent. There's not much financing going that way. They've actually won over heart diseases based on the fast reaction time, but not because of genetics. Still, we have high hopes for the progress in that area. Thank you. Thank you, Maria.